Some color, very strange. McKenna, can you hear me all right?
Yeah, I can hear you. Thank you.
Okay. You're going to be joined by at least 2 other people. So you won't be alone.
Okay.
Good gravy. Where is everybody? Oh, here come 2 more. Emily, should I take from the fact that you're here that you guys lost? That's a sucky draw you guys got. So otherwise you wouldn't be here. You'd be playing the next team, right? Oh, Wednesday. All right, so you would have been here anyway. All right.
Yes, but did you do it in Google Docs? Yeah. So you have to download it?
It's a significance of.
I don't know. She's okay with the switch. Yeah, I mean, obviously. She's okay. Yeah.
All right, it is time to start. Hope you guys had a chance to enjoy the remarkable weather we had this weekend. It got warmer. So I had an experience I've never had before. I received a text from my youngest daughter. You have to call us. You have to call us. You guys have to call me. So we called her. There she was with her boyfriend with a ring on her finger. My youngest daughter's engaged. So I get a son. After 3 daughters, I finally get a son. So yeah, so it will be 2023. And Brady's a good guy, comes from a great family. And as she put it, it's easiest yes I ever had to say. So that's great to hear. I'm very happy for them. And yeah, so again, never had that experience before. So there's some things you just wish they just kind of last so you could kind of savor them. That was one of them. Very good. So well, maybe your weekend wasn't quite that eventful, but hope you guys enjoyed it. All right. Getting back to the subject at hand, we were looking last time at the receptors cells use to bind to things that they're going to phagocytize. So with phagocytosis, the phagosome engulfs the particle, the cell, the virus, or whatever, and destroys it. It surrounds it, it binds it to its membrane, it ruffles its membrane and brings that membrane, bit of the membrane that the thing is bound to, into the cell in the form of a phagosome. That phagosome then fuses with a lysosome, well, several lysosomes. And the lysosome contains enzymes that make reactive oxygen species. The biggest culprit being myeloperoxidase. That is found in neutrophils. It's not found in macrophages, and a whole host of enzymes that take the particle and disassemble it, destroy it. So you have reactive oxygen species that kill the bacterium, virus, whatever. You have enzymes that take it apart. This binding on the surface that lets the phagosome gobble it up, these are pathogen recognition receptors, or PERs. The cat says purr when it gets the mouse. So think of this essentially as teeth that it can sink into the mouse, if you will, because the cat says purr, so teeth. And in your body, there are proteins that are able to bind these receptors. These proteins bind to the surfaces of pathogens. They're called opsonins. Opsonins. Opsonins bind to the surfaces of pathogens. They make phagocytosis of that pathogen much more easy for the phagosome. So when I was a kid, I worked on my dad's farm. There were these devices that we used to use to get the boxes off the truck, and then boxes full of grapes from the field, and then get them into the packing house. And the conveyor belt, very often when you put a box of grape on it, the conveyor belt would do this. So we have these large, I think they were either lead or they were pig iron weights that we put on the bottom of the arms of the conveyor belts. These things were round and they were flat. And when they were on the ground, first of all, they were heavy because they were solid metal. And plus I was a kid, so I wasn't very strong. Trying to pick them up, you had to somehow wedge your fingers under them. And they're on a flat floor. To pick them up was not an easy task. So the workers used to pull out their pocket knives and stick a knife under it and use the knife. They carry these big, beautiful pocket knives with very strong blades. They'd wedge their pocket knife under it and pick it up and then grab it skillfully without cutting themselves. If I would have tried that, I'd have cut myself in order to pick it up. So opsonins essentially act like that. They act like a place where you can grab onto the thing and take it up. Otherwise, you're trying to pick up a Chinese water chestnut with a pair of chopsticks. It just kind of skirts around the plate and you never quite get it. So they're handles, if you will. They're handles. So opsonins bind to these things and they create handles that the phagosome can use to take it up and destroy it. So some of these receptors that bind to various molecules on the surfaces of pathogens, there are several of these, but the ones that are probably the most important for our purposes. So CD14 binds to lipopolysaccharide. Lipopolysaccharide is found in the outer membrane of gram-negative bacteria. So CD14 binds to lipopolysaccharide, and CD14 is found on the surfaces of phagosomes. They use that to bind to gram-negative bacteria and gobble them up. And these receptors are all in this greater category of scavenger receptors. There's another receptor called scavenger receptor A. Scavenger receptor A. And it was called scavenger receptor A because when it was discovered, nobody knew what it did. So they called it, oh, it's a scavenger receptor, because it was pretty clearly a protein that was embedded in the membrane. So it had to bind something. So they said, oh, it's a scavenger receptor. And it's the first one of its class. So let's call it scavenger receptor A. Well, the name stuck, even though it's a stupid name. Scavenger receptor A binds lipopolysaccharide, but it also binds to teichoic acids. Teichoic acids are in the cell walls of gram-positive bacteria. So this one binds to the cell walls of both gram-positive and gram-negative bacteria. Scavenger receptor A. Then there's another one that has a much better name called Marco, like Marco Polo. In fact, the person that discovered it named it Marco Polo because they like to play Marco Polo with their kids in the pool. And they said this thing plays Marco Polo with bacteria because it binds to bacterial surfaces. What it precisely binds in the bacterial cell wall is multifaceted. So it binds bacterial surfaces. And this Marco is also found on the surfaces of phagosomes. Then you have a variety of other receptors that bind several other things. So Minkle binds to a specific lipid that's found in the cell wall of mycobacteria. This lipid is called cord factor. The more cord factor a mycobacterium has, the more pathogenic it is. And Minkle binds to cord factor and helps it gobble the cell up. Finally, dectrin. Dectrin 1, there's a family of dectrins, but dectrin 1 binds to beta glucans. Beta glucans are found in the cell walls of fungi. Some capsules of bacteria will be made of beta glucan as well, but usually you find beta glucan in the cell walls of fungi. Now, there's another thing that your immune system looks for, and that is the sugar mannose. Mannose is a 6-carbon sugar. Mannose is used relatively sparingly when it comes to our own biochemistry. We do have glycoproteins with mannose in it, but not very much. Fungal cell walls have lots of mannose in them. So if your body comes across something with a lot of mannose, your body says, that's not me. And so mannose turns out, if there's lots of mannose, that turns out to be a signal that something is awry. So mannose is found in fungal cell walls. It's also found in viral glycoproteins. And it's found in bacteria as well. So large amounts of mannose in general are Nixay, shall we say. And there's a protein that's called the mannose binding lectin. It's a big protein, binds to large sections of mannose, and then on the surface of macrophages, there's something called the macrophage mannose receptor. And the macrophage mannose receptor binds to the mannose binding lectin. And it also binds to large amounts of mannose. So essentially, these cells have lots of ways to recognize bacteria and phagocytize them. And when they phagocytize those cells, they kill them. And when they're active, when they're phagocytizing cells and they kill them, they will release cytokines. And those cytokines will recruit other white cells to that area. The other thing that happens in areas that are infected is that tissue cells called mast cells release histamine. Histamine is also released when tissue is damaged. So let's say Brianna back here, let's say she jumps for the ball because she's a goalie, lands on the ball, but then also gets a nice cleated foot right in her side, which I'm sure has probably happened more than once. After she says, ow, and I hope that kicker gets a nice yellow card for that, but knowing the refs we have in this league, they probably not. The next day, she's probably going to be a little black and blue where she got the foot in the side. What's that black and blueness from? Well, let's take it back to the game. When that happens in the game, she's going to rub her side. And if she were to look at it, it'd probably be red. The redness is from damaged tissue cells. Those tissue cells are damaged, they release histamine. Histamine causes capillaries, causes the endothelial cells of capillaries to contract. When they contract, the capillaries become leaky. They become leaky, they start to leak fluid into the area. Leaking that fluid into the area causes the area to swell. You swell the area stretches, the stretching causes pain. The leaky capillaries also expand, which increases their leakiness and increases blood flow through the area. That causes the area to turn red. And the increased redness, increased blood flow, and increased red cell arrival produces heat. So now you have the 4 signs of inflammation: redness, pain, swelling, and heat. Redness, pain, swelling, and heat, which you easily remembered if you use the Latin terms: rubor, calor, tumor, and dolor. And if you speak Spanish, you'll recognize these 2 terms very quickly because the Spanish words are pretty much the same. So rubor, redness due to the vasodilation. Calor, due to increased blood flow and increased metabolism of the area. Tumor, which originally just meant swelling. That's due to the leakiness of the cells causing increased fluid and swelling of the area. Dolor, that's due to stretching of the area, and histamine sensitizes pain receptors. take your vitamins. Inflammation can lead to loss of function, but that depends upon the level of inflammation, doesn't it? Will Brianna get up and just keep playing? She'll get up and just keep playing. Now, if she gets kicked 27 times in that game, all over her body, she's probably not, especially in the head, she's probably not gonna be able to function. My first question would be, where's the ref in that case? Because the ref is clearly not protecting the goalie. But inflammation can be systemic, in which case it could lead to very definite loss of function, or it can be local. And if it's local, very often it hurts, but it doesn't lead to loss of function. And inflammation very often is due to the innate immune response, but it often kicks off and leads to the beginning of the adaptive immune response, which will be the next subject for us. Now, the goal of inflammation is to kill whatever is invading your body. That's the main goal. The second goal is also to get rid of the debris generated by tissue damage. Why do you want to do that? Bless you. Sorry? No. Let's say a tornado, Natalie, let's say a tornado hits your house. Insurance will say, we'll rebuild your house right on the site. Okay, what do you have to do first before you start rebuilding? Get everything out of the way. That's what your body's doing. It's getting everything out of the way so it can heal the damaged tissue. So phagocytes will not only clean up the bacteria, they also clean up the debris. Brooke throws a shot put. Let's say Brooke hyperextends one day with that shot put. Ow. Oh, my teres minor. Ow, 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 ow. That tendon now essentially got stretched. Some cells went pop, essentially. Is there infection in that tendon? No. But does she get redness, pain, swelling, and heat? Yeah, that's part of the reason why the trainer will ice it to decrease the damage, because now red cells will come in, they'll clean up the debris so that the tendon can now heal. That's why she'll be asked to take it easy and not throw. The shot put you throw is 12 pounds? 8 pounds. Okay, 8 pounds. The guys throw 16. Freaking bowling ball. So she'll be asked to, do a golf ball or something like that, or a softball or something like that instead of an 8-pound shot put while it heals. So cleaning up the debris is important too, so that healing can start. So inflammation is the initiation also of the healing process. So inflammation occurs in stages. They're nicely elaborated here. So you have an initial response where you have the release of various chemical mediators like histamine, like other types of damaged cells that release cytokines and what have you. Histamine is vasoactive. White cells come in, they start to clean up the mess. Then you get a so-called vascular reaction. That vascular reaction involves the vasodilation. In damaged tissue, you'll actually start to make new blood vessels. That's called granular tissue, full of these new blood vessels. Those new blood vessels are meant to flood the area with oxygen so that the tissue can divide and regroup. If you have infection, you'll get pus and what have you, exudate. Exudate is the fluid that's made. Oh, another group of molecules made during inflammation. I didn't mention those. They're made locally, they're made by damaged cells, and they're made by white cells. They don't diffuse very far from the area. They're mainly involved with local inflammation or molecules called prostaglandins. They will circulate systemically, but they don't have long half-lives. Prostaglandins are made constantly throughout inflammation, and quelling prostaglandin synthesis can quell inflammation pretty effectively. If you guys are familiar with a group of drugs called the non-steroidal anti-inflammatory drugs, of which aspirin and ibuprofen and naproxen sodium, Nuparin, drugs like that are all part of, that's how they work. They prevent the synthesis of prostaglandins. So those are another one of your vasoactive mediators. So exudates that you get in inflamed areas, they're sometimes pus, but sometimes they're just the fluid that comes from the leaking vessels. After the cells and the debris have all been cleaned up, the tissue now will start to heal. Typically, small areas heal very quickly and without scars. Larger areas, we usually need a scar because something that needs to happen to heal, the tissue needs to be brought together in order for you to get so-called epithelialization. If it's too far apart, now you have to make a collagen bridge. And the scar is made of collagen. You have a question?
Yeah. What would it have to do
If your body did not induce inflammation, you'd be subject to infection. You'd also have trouble healing because inflammation kicks off the healing process. So both of those things would be necessary. But both of those things require inflammation, rather. Inflammation is necessary for both those processes, fighting infection and healing. So if you get, say, a splinter, which working on a farm around a packing house where everything was made of wood, I've had more splinters than you can ever dream of. Splinters, if you don't deal with them right away, they get infected. That happens. And it gets big and red and indurated. And then you try to pull the splinter out and all this pus comes out. So the rule was with a splinter, once you get a splinter, you deal with it right away. That way you don't have the issues later on. So with a splinter, if you had a splinter and you didn't realize it, you'd have local infection. That infection might become systemic. You might become septic just from a splinter because you couldn't fight the infection locally. That's bad, right? So inflammation, as much as we hate it, and as much as it can really hurt you, because with inflammation comes not only damage to the bacteria, but the local tissues also take damage because the white blood cells, they're very sloppy when it comes to eating bacteria. And some of these digestive enzymes, they leak out. Some of the reactive oxygen species, they leak out. So they damage nearby tissues. That's why long-term inflammation, chronic inflammation, can be so damaging to tissue. And that's why autoimmune diseases that are characterized by chronic inflammation can cause lots and lots of scarring, like scleroderma, for example, systemic sclerosis. That's essentially a hallmark of that disease.
So like a lot of something like super inflamed.
Yeah.
Like overworking.
Which is what happens with COVID-19, essentially. You get this massive, so-called cytokine storm, where the cytokines that are the pro-inflammatory cytokines that are produced just crescendo, if you will, and they keep going, and you have this massive amounts of systemic inflammation, and all your vessels are leaking. So your blood pressure crashes and you go into shock, essentially. Now, there are some cytokines that are released that are meant to essentially keep inflammation within certain boundaries. For example, interleukin 10 is a regulatory cytokine that's meant to downregulate inflammation. If, though, the other one is TGF beta. So that one, remember that one by, thank God it's Friday. Thank God it's Friday. Thank God it's Friday and I'm not, I don't have any more inflammation. So that one takes the inflammation knobs and turns it down as well. Those down regulatory events essentially get overwhelmed by the massive amounts of inflammation in some diseases. Toxic shock's another example. And that's due to these things called superantigens, which we'll look at a little bit later, that take the inflammation and just turn the knob all the way to the top and then essentially pull the knob off so you can't turn it down. That sort of thing, essentially, that's what causes the cytokine storm. And that can lead to patients' deaths. That's why these drugs like tocilizumab and now sarilumab, which bind and prevent interleukin 6. Interleukin 6 is one of the first interleukins made during inflammation. Interleukin 1 and 6 and tumor necrosis factor, those are the three acute phase proteins. By turning that one down, you essentially prevent, you jam the on knob, essentially, so that inflammation gets stuck. So then these down-regulatory molecules, they now take over and inflammation now starts to get quelled. I kind of rambled. Did I actually end up answering your question, that ramble? Okay, good. Macrophages are some of the biggest cleaner uppers, if you will, of this. Was where neutrophils come in, burn their candle at both ends, do their damage, and then they die. Macrophages are longer lived. They're the more reserved. They come in, and then macrophages actually later on in the inflammatory response become become cells that are less pro-inflammatory and more pro-healing. And consequently, there's two main types of macrophages you'll see talked about in books, the so-called M1 macrophage. And if you are into weapons at all, the M1 is the carbine. The M1 is a pro-inflammatory macrophage, so that's that one. The M2 is a more pro-healing macrophage. And so during inflammation, the first macrophages that get there, they're M1s. Then as inflammation starts to be downgraded, the M2s take over, they downgrade inflammation and they promote tissue healing. We talked about chemotaxis. Chemotaxis is this tendency for white blood cells to move towards areas of inflammation, toward areas that are affected by bacteria, toward areas where cells are damaged, and what happened. That's chemotaxis. Diapedesis. Diapedesis refers to the ability of several types of white blood cells to flatten and then move in between the endothelial cells of various blood vessels to get into the peripheral tissues, in particular capillaries. So they move through the endothelial cells, they attach in very specific, there's a lot of cell biology involved with that, but for our purposes, they attach loosely, they then roll. They attach loosely and then they roll across the cell, and then they flatten out so that they're flat as a pancake. The cell, because the attachment now loosens its connections with the other cell, and then they slide in between those two connections. And that's how they get to the peripheral tissues from the bloodstream. Diapedesis. And this slide is meant to essentially show how essentially these cells attach, roll, signal, and loosen their connections, and then they flatten and then go through. I wish this would have shown it flattening, but well, anyway. Here it is essentially being caught in the electron microscope. You can see that the endothelial cells have loosened their attachment to each other. This cell now is going through and it will go to the other side of the peripheral tissues over here. Blood vessels right here. And you have two cells that are in progress there. Neutrophils, when they get to the site of inflammation, by damaging tissue, they will also institute the clotting pathway in many cases locally. That's a good thing. Why is it a good thing? Because it walls the area off. Those bacteria now are trapped. They're trapped with a bunch of white blood cells. So if you wanted to hurt somebody, you throw them in a cage with a gorilla, and then you close the door and you lock it. That's essentially what your body is doing to these bacteria. They're trapping them in a cage with cells that are specialized at one thing, killing them. So you trap them in a cage with a bacterial assassin. That's what neutrophils are. Anything that generates pus or that generates a fever is called pyogenic. That's what pyogenic is. Again, those neutrophils that come in, they work hard, kill, they die. And the exudate from the vessels, the dead neutrophils, and the dead bacteria, that gemisch, nice Yiddish word for you, that gemisch is called pus. Pus. And the fever is caused by interleukin 1, the cytokine called interleukin 1. It signals to the hypothalamus and it raises the body temperature. Several experiments have shown that fever actually is good for your body when it comes to infections. If you take two groups of people, have one group take the cold head on and not try to drop the fever, have the other group take medications to drop the fever, the group that doesn't drop the fever, they get over the cold faster, consistently. So fevers do tend to help you fight infection better. After I told students this, one student essentially made a resolve to never take aspirin or ibuprofen ever again. I wouldn't say that. If you're really sick and you're miserable, yeah, you can go ahead and take aspirin and ibuprofen. But on the average, you'll tend to get better faster if you don't. Rebecca, was that a question or no? Scratch it. Okay. Some bacteria and viruses and fungi and other organisms make things that also cause the hypothalamus to reset the thermostat. So those things are pyrogens. They're exogenous pyrogens. They're made by other organisms. LPS, lipopolysaccharide, is an exogenous pyrogen. That's one of the reasons my patients who are septic with a gram-negative infection will have such high fevers. When we look at typhoid fever, fevers of 104 are not at all uncommon. And often the fever will kill the patient before anything else does. So these things that are released by cells that drive up the fever, those are exogenous pyrogens. Things that are released by your own white blood cells, like interleukin 1, interleukin factor, that raise the body temperature, those are endogenous pyrogens. And fever, again, fever prevents the multiplication of several different pathogens, well documented. Interferes with iron acquisition by bacteria. Increases metabolism, so the white cells now get more active, more able to kill bacteria and viruses. Increases the efficiency of phagocytosis, increases hematopoiesis. Fever, from the perspective of fighting an infection, is good, even though it makes you feel miserable. The other thing fevers do, what do you want to do when you have a fever? Natalie, when you have a fever, do you feel like going to the gym and doing your workout? No, what? No? What do you want to do when you have a fever? Oh, lay down and get under blankets. So you want to take it easy so that your body can concentrate its energy towards fighting the infection. That's the other thing fever does. It causes you to want to rest. And by resting, all those bodily reserves get channeled towards fighting the disease. So sometimes one of the best things you can tell your patient to do is go home and rest. And if your patient is one of those who feels guilty when they lie down, like my wife, tell them this is not a competition. One thing I didn't mention. We're going to look at, when we talk about adaptive immunity, at these proteins called antibodies. Antibodies are extremely good opsonins. In fact, they're one of the best opsonins your body has. So antibodies, very good opsonins. A slight fever makes you tired and makes you miserable. It's not life-threatening. However, high fevers, like the kind Jenna was referring to, then cause your heart rate to go up pretty high. They can cause your respiration rate to go up pretty high. And they can also lower the seizure threshold. So fevers that get too high are counterproductive, aren't they? For those reasons. And if you have a patient who spikes a fever, when you work in the ICU, there'll be the 5 reasons someone can have a fever, and you'll learn these: wind, water, walking, wonder drug, so on and so forth. And wind, that's pneumonia; water, urinary tract infection; walking, the patient hasn't gotten up and walked around like they're supposed to, so they're splitting a fever. Wonder drug, some drugs will cause a fever and what have you. So the 5 W's, those, and 3 of those are infections. So those are some of the things you look at if a patient has a fever. High and prolonged fevers can be inimical to the cardiovascular system. That's just something to keep in mind. All right. Are there any questions before we look at some antimicrobial products, interferon, and then complement? All right, so interferons. There's 2 types of interferons. One of them is made to signal to other cells, help, I'm being infected by a virus, modify yourself so that it's harder for viruses to infect you. So one group of interferons, the alpha and the beta interferons, are made by cells that are infected by viruses, and they tell other cells, watch out. Do something to your machinery so the viruses have a harder time infecting you. Gamma interferons are made by T cells, T helper cells in particular, and they activate other immune cells, in particular macrophages. So think of interferon gamma as a candy bar for macrophages, or a power bar for those of you who eat power bars after or before workouts or something like that. Those are interferons, powerful signaling molecules. They're used for some viral infections or recombinant versions that are given for some viral infections. They stimulate phagocytosis. When viral infected cells secrete them, they cause other cells essentially to degrade viral RNA, and they prevent, they modify those cells when they get these interferon signals, they modify their machinery so they won't translate viral RNAs into proteins. Those are the characteristics of interferons. Okay. Oops. I know what I did wrong. I know what I did wrong. I know what I did wrong. All right, so back to here. Okay, got to do this in order or it won't show it. All right. Good night. All right, listen to this video.
When a cell is infected by a virus.
Oh dear. Oh, you did that. I thought, oh gosh, did I blow something? Yeah. What is up with this?
Virus. The virus and
Okay, yeah, that works. Let's try that.
when a cell is infected by a virus, the virus enters the cell and creates instructions that are not. The cell
All right, so thank you, Jenna. So those are essentially the biological effects of interferons. They're very powerful signaling molecules and they're used extensively both during viral infections, and then gamma interferons are used very extensively if you have intracellular parasites of some sort. And this leads us now to complement. So for complement, would you please take this handout? And for those of you on Zoom, I'll put this handout online. So, and I'll hold it up to the camera here so you can see it. So what is complement? Complement is a cascade of reactions initiated by proteins that are in your bloodstream. These proteins are made by the liver. They're just found in your bloodstream. They're inactive unless you get an infection, then they become activated. Complement proteins are known as capital C and then 1, 2, 3, 4, 5, 6, 7, 8, 9. So they're given names only a biochemist could love. So if you look, please, at the handout. Let's go through essentially there's two pathways. There's a classical and an alternative pathway. So the first component of the classical pathway is a big protein called C1. C1. If you have an organism bound by antibodies, and those antibodies are close by, C1 binds to those antibodies. So C1 is not activated unless you have something bound that is bound by antibodies. So already, they're not activated unless the immune system itself is wise to the invader. When C1 binds to antibodies, it changes its 3D shape. And now it is able to be bound by a different complement protein called C4. C4. The binding of C4 activates C4, and C4 now becomes a protease. What do proteases degrade? Proteins. C4 is a protein, and it's one of those rare proteins that degrades itself. And the fragment that it makes is called C4 lowercase b. C4 lowercase b, and C4's lowercase b does two things. It sticks to C1, and it also sticks to the surface of the microorganism. All right, you with me so far? This C4b protein bound to C1 is then bound by another complement factor called C2. C2. So my immunology professor used to say, the beginning complement factor, remember, 142. Because it's C1 and then C4, then C2. 142, that tells you the sequence right there for the classical complement pathway. C2 is clipped by C4 into 2A and to C2A and C2B. C2B binds to C4B. And the C4B, C2B complex is known as a C3 convertase. C3 convertase. It's on your handout. C3 convertase binds the complement protein C3 and clips it into two fragments, C3A and C3B. C3b has an uncanny ability to bind to the surface of bacteria. And C3b is going to be a very important opsonate because white blood cells have C3b receptors. C3b also will bind to the C3 convertase, and now it becomes a complex of C4b, C2b, and C3b. So we have C, 2, 3, and 4 essentially, B. That complex is called the C5 convertase, the C5 convertase. That convertase will take the complement protein C5 and clip it into C5a and C5b. C5a is a very important protein because it can stick to the surface of cells, and it becomes an important opsonate. However, C5a is also a very potent chemotactic agent for white blood cells. So circle C5a, and right next to it, chemotactic agent. If you isolate C5a in culture and give them C5a, they go nuts. Give me more of that, give me more of that, give me more of that. Where to go, where to go, where to go, where to go. And you can make a little breadcrumb trail of C5a and they'll follow it. Just like Hansel and Gretel and the trail of stones to get out of the forest. Or was it breadcrumbs? I can never remember the story. Anyway, C5b now is a assembly point for C6, 7, 8, and 9. C6, 7, 8, and 9 assembles into the membrane of the cell and forms a pore. The cell now starts to leak. And everything inside the cell now leaks to the outside and the cell dies. And C5b and C6, 7, 8, and 9 constitute the so-called MAC. No, that's not mac and cheese. That's membrane attack complex. Membrane attack complex. So what does this culminate in? It culminates in the death of the cell. Is this complicated? Yes, it is. I will make no bones about how complicated this is because it is complicated. But the thing I want you to see is that it starts with antibodies bound to an invader. It culminates in the formation of the membrane attack complex that pokes a hole in the membrane and kills the cell. That's the big part. And you make things along the way that facilitate the immune response. You make opsonins, important opsonins, and you make an important chemotactic factor, namely C5a. And then C3b is important opsonin. And yeah, what have you. Right, any questions about the classical pathway? Now let's talk about the alternative pathway. There's two ways to do the alternative pathway. First of all, this protein here, mannose binding ligand. Mannose binding ligand molecularly has a 3D structure that's almost identical to C1. Except mannose binding ligand will bind to the surface of mannose-rich cells, like fungi, viral glycoproteins on the surfaces of enveloped viruses, and some bacterial cell walls. And then it's bound by C4 and blah, 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 and so on and so forth. So in that case, it's the classical pathway. You just substitute the MBL for C1. Cassidy, I saw that. Now, the actual alternative pathway starts with C3. C3 has the tendency to just automatically clip itself from C3b, C3 to C3b and C3a, but it doesn't last very long inside the bloodstream and it's not of any consequence. However, if there are bacteria in around, clipped C3b will bind very tightly to the surfaces of bacteria. And if it does, then it recruits another protein in the blood that's called B. It's just called factor B, capital B, that's all it is. And that clips it into big B little a and big B little b. Who names these things? Those make a complex, and that complex now becomes a C5 convertase. And so you convert C5 into C5a and C5b, and C5b now assembles C6, 7, 8, 9 into the MAC that degrades the cell. That's the alternative pathway. So you skip all this C1, C4, C2, you skip all that bishwhack. You just go pass go, collect $200, go straight to C3. And that is the complement pathway. So the alternative pathway does not require antibodies to get started. It's initiated by the presence of foreign cell antigens, and it's quicker than the classical pathway. Brooke?
What's the example of when cells are used for classical versus
So certain cells, for example, that have capsules that can escape the original antibody response also, they have trouble being eaten by phagocytes. The classical pathway, the alternative pathway becomes extremely important in fighting encapsulated bacteria. And here's a cartoon version of that and what have you. Okay, and you also have in your body some antimicrobial peptides. These also can put holes in membranes, and they're called defensins and things like that. They essentially just put little pores in membranes and they make membranes leaky. They damage them and things like that. And then the book asks you essentially to put the steps of phagocytosis in the correct order. So which of those comes first? Second first? Second first? No, that's at the end, isn't it? Second first? How about that? Yeah, you gotta get there first. You gotta get there first. So 6 is going to come first. Yeah, that's gonna go first. Okay, what's gonna come next? That one? No, you have to bind to it first, don't you? That's gonna come next. What's gonna come next? Engulfment. You ruffle the membrane, you bring it inside, you engulf it, then you form the phagosome. Then you form the phagosome. And what comes next after that? You form the phagosome, you fuse it with a lysosome and form phagolysosome, then you kill and destroy the bacterial cells, then you release the residual debris. So this one. D is the correct answer there. Very good. All right. Let's take 5 minutes. Rest your brains a little bit. I threw quite a bit at you, but don't worry. It's about to get even more complicated. And we'll start in the next chapter. So, ooh, we lost one. We lost McKenna. Where'd she go? Yeah. I'm sorry, too. Okay, there we go. Okay. Okay, let's resume. So for those of you on Zoom, the handouts are all in a shell under course basics that says handouts, class sample handouts. All right, let's look at the adaptive immune response now. And for this one, you will need to have this handout at the ready at some point. So adaptive immunity is going to marshal your B and T lymphocytes. Your B and T lymphocytes essentially go under a highly selective process that sieves them to make sure that they don't react with your tissues. Each each mature B and T lymphocyte will be able to react to one type of antigen. And so the ability of your body to react with different types of foreign antigens, foreign substances, that condition is called immunocompetence. Immunocompetence. If someone is not able to react to countless foreign substances, they're called immunodeficient. Immunodeficient. And if their condition is due to medications or diseases that prevent them from being immunocompetent, that's called immunosuppressed. Immunosuppressed. We talked about antigens before. Antigens provoke an immune response. They're seen, they're identified by the immune system as foreign, and your immune system reacts to them. Yes, our bodies contain things that are potentially antigens, but our immune system is made to tolerate them. Now, you have things in your body that your immune system will react to, but your immune system doesn't react to them because it never sees them. Case in point, the protein in the lenses of your eye. We can take lens protein, we can inject it into your body and get a beautiful, robust immune response against it. Why doesn't your immune system respond to your lens protein? Because your immune system never sees it. The lens protein is socked away inside your eye. The eye essentially is privileged, it's sequestered away from the immune system. So your immune system never sees lens protein. There are other types of proteins in your body like that. They're secunded, they're sequestered away from the immune system. Those types of sites are called immunoprivileged. Immunoprivileged. So the adaptive immune response is both specific and it, like an elephant, has a long memory, it remembers. If it sees that antigen again, it can call up a response against an antigen that's specific very quickly. And that, dear ladies, is the basis behind vaccination. This is just from your book. I didn't like this picture. So specificity, diversity, there will always be a diverse response to the antigen. It's inducible, it's clonal, there's tolerance in that it doesn't react with self-antigens, and it remembers. It remembers. That picture, if you were to tape that on your refrigerator and look at that occasionally, that definitely wouldn't hurt you. Each sets of lymphocytes, B and T cells, undergo a very strict developmental regimen to ensure that they don't react with self-antigens. If they do react with self-antigens, they undergo programmed cell death. So all lymphocytes arise from the lymphoid progenitor. After the hematopoietic stem cell divides, that cell becomes a lymphoid progenitor. There's good evidence for the existence of the lymphoid progenitor. The myeloid progenitor, there's good evidence that the myeloid progenitor doesn't exist. Instead, that cell goes straight into some sort of myeloid lineage. There doesn't seem to be I know books keep showing it divides, there's a myeloid progenitor and a lymphoid progenitor. Well, people have tried very, very hard to find the myeloid progenitor, both in the mouse and in humans, and they haven't been able to find them. That's led several people, me included, to say it doesn't exist. But the lymphoid progenitor does exist. So those cells are they're born in the bone marrow. The T cells, however, will leave the bone marrow and will go to the thymus. So they undergo their T cell education, if you will, in the thymus. The B cells will undergo their education in the bone marrow. So if we look at B cell maturation, B cells in the bone marrow will attach to stromal cells in the bone marrow. Those stromal cells will release interleukin-7. That's very important for the continued growth and maturation and survival of the pre-B cell. If you don't have interleukin-7, you will not have B cells. Or if you don't have an interleukin-7 receptor, your whole B cell lineage gets deleted, you don't make antibodies. Very bad. Very bad. During that time, the stromal cell is going to present the B cell with lots of different self-antigens. And the B cell is going to start to express on its surface the beginnings of an antibody. If, as it goes through that set of pre-antibodies and then finally more mature antibodies, if any of those react with any of the self-antigens that the stromal cell presents to it, the stromal cell withdraws interleukin-7 and the pre-B cell dies. That's how the bone marrow prevents the release of B cells that self-react. Once, however, it makes a proper surface antibody, and that surface antibody is called an immunoglobulin M antibody, and it doesn't react with self-tissue, it will be released. And it will go usually into the bloodstream and then usually into the lymphatic system and typically take up residence in a lymph node. But some of them can be circulating in the bloodstream. And if it's activated, it will undergo so-called, well, first of all, it will start to secrete antibodies. When it starts to secrete antibodies, it becomes known as a plasma cell. So plasma cell is a mature B cell that's been activated. There's a term, it's not used so much anymore. Once the B cell is released and it's not activated, in the literature it used to be called a virgin B cell. And it's not called that anymore. Now it's called a naive B cell. Just a B cell that's not been activated yet. Whatever you can call it Frank or Joe or Bob, who cares? But it's a B cell that's not received the signal to start making antibodies. Once it does, notice it goes from a really round cell to something that looks more bean-shaped. The amount of endoplasmic reticulum goes way up because it's making a secreted protein, namely an antibody. It's making lots of it too. The other thing these cells do is they divide. Activated B cells, activated lymphocytes in general, divide. And they divide like mad. They become plasma cells, and then if these keep getting activated, they will undergo something called isotype switching. And they'll make a different type of antibody other than an IgM. That's the B cell story in a nutshell. Now let's look at the other side, the T cells. T cells leave and they go to the thymus. They go into the bloodstream, they enter the thymus through these little capillaries that are called high endothelial cell capillaries. They get into the thymus. When they get into the thymus, they don't express any cell surface proteins that will really distinguish them as T cells. In the thymus, they start to make their T cell receptor, and then they start to make the proteins that associate with the T cell receptor. There's two proteins that associate with the T cell receptor, that can associate with the T cell receptor. There's a protein called CD4. There's another protein called CD8. So in the first stage in the thymus, the T cell is trying to make a proper T cell receptor, and it doesn't express either CD4 or CD8. At this point, the cell is called a double negative cell, a DN cell. Once it makes a proper T cell receptor, then expresses both CD4 and CD8 and becomes a so-called double positive cell. If at any time the T cell receptor binds to self-antigens, the stromal cells of the thymus will induce that cell to undergo programmed cell death. Over 90% of the T cells that get to the thymus will die because of the stringent self-selection process. After becoming a double positive cell, the double positive cell will try to recognize different types of molecules presented to it by the stromal cells in the thymus. If it is able to recognize these proteins in combination with CD4, then it keeps the CD4 and it dumps CD8 and becomes a CD4 positive T cell. And CD4 positive T cells are T helper cells. If, however, it binds to the molecules presented to it by the stromal cells in combination with CD8, then it keeps expressing CD8 and it dumps CD4 and becomes a CD8 positive T cell. And this becomes a T cytotoxic cell. And it goes from being a double positive to a single positive, either CD4 or CD8 T cell. So now this T cell has a proper T cell receptor, and it's associated with either CD4 or CD8. Then it gets released into the bloodstream, and it will usually go straight to either the spleen or lymph node. You don't have a lot of T cells circulating in the blood. Is there any piece of that you want me to repeat that wasn't clear? The upshot of this is that if you react with self-antigens, you get the deep six. You are given signals and told to, essentially, to off yourself, if you will. Now, this stringent process, is it perfect? No. There are people walking around on this green earth with autoimmune diseases. So is it a perfect process? No. but it's pretty stringent. And that's the reason why most people who get autoimmune diseases don't get them until later in life. Because as these cells adapt to different infections, now they may end up reacting with self-antigens, but they don't usually get them early on. There is a genetic condition called DiGeorge syndrome. Where you don't have a thymus. No thymus, no T cells. No T cells, all you have now is innate immunity. Without T cells, you can kiss adaptive immunity goodbye. And people with that kind of genetic condition are severely immunocompromised. Because as we'll see, B cells and T cells work together to form the innate immune response. All right, pause, look for questions. You guys are acting like I've tortured you today. Hung you by your thumbs and poked you with a hot iron. Jenna?
In our eyes and other parts like they can't see them, so they can't destroy them. Is there any case where that's happened to somebody where they can't see them?
Yeah, severe trauma to the eye that causes blood to leak into the lens cavity will cause an immune response against the lens cavity in one eye that might spread to the other eye as well and might cause you to become blind. That's why if you have damage to the eye, you have to stabilize that eye as fast as possible. And that's why if the eye just can't be saved, it'll be surgically removed for that reason, because you'll compromise the other eye if that happens. Because now the immune system is seeing lens protein, and it will not be denied. All right. So how do you tell self from non-self? The answer is barcodes. Barcodes. Sydney's looking at me as if to say barcodes, huh? What? So when you go to Meijer or Walmart or whatever, you buy your coffee, let's say for the week, or I don't know, the way some of you drink, maybe for the day. You give it to the nice person at the checkout and they slide it past the scanner and the scanner goes beep. It recognizes that code. Your body has a series of surface proteins that act like barcodes. Those surface proteins are encoded by a gene complex called the major histocompatibility complex, or MHC complex. This complex is highly polymorphic. Who knows what polymorphic means? Changes shape. In this case, polymorphic means that these genes vary wildly from person to person. You have multiple alleles. When you hear allele, think of type of that gene. So I could have, for example, 7 sheets of paper and they're all different colors. So we have white paper, we have black paper, we have orange paper, we have blue paper, we have purple paper, we have brown paper. Are they all paper? Yeah, but they're different types of paper. They're different alleles of paper. So these genes make the same cell surface proteins, but they're encoded by different alleles. In fact, multiple alleles, hundreds of different alleles in some cases. That's why these genes vary wildly. And the proteins they encode are called HLA proteins, human leukocyte antigen proteins. Human leukocyte antigen proteins. And there's two main groupings of HLA proteins, class 1 and class 2 HLA proteins. Class 1 HLA proteins appear on the surfaces of all nucleated cells. Class 1 HLA proteins are found on the surfaces of all nucleated cells. Class 2 HLA proteins are found on the surfaces of specific white blood cells that present antigen to the immune system. Present antigen. What do we mean by present antigen? In order for the immune system to respond against an antigen, the antigen has to be presented to them. This presentation of antigen licenses the immune system to respond to it. Do you see how heavily regulated this response is? But you see, it has to be this heavily regulated. It has to be, because the immune system is like a caged animal. It's like, have you seen the Tasmanian devil? Okay, good. The Tasmanian devil. The Tasmanian devil is always in this cage. We let him out of this cage and the whole set gets destroyed. That's what your immune system is like. And so the key to unlock the Tasmanian devil, which is the immune system, is antigen presentation. Except the Tasmanian devil is not going to destroy the whole set. The Tasmanian devil is going to recognize that one thing and destroy that one thing when it comes to the adaptive immune response. But in order to get a permission to do that, the antigen has to be presented to it. And only cells that express these class 2 MHC genes that express these class 2 HLA proteins on their surfaces can present antigen. So here is the structure of a class 1 MHC gene product and class 1 HLA protein. So it essentially forms a little cup, and it has a little piece that then sticks to the membrane. And it forms a cup. This cup is right here. That cup is going to bind antigen. The class 2 is a two-partite thing, and it has, as it says, another cup. That cup is going to bind antigen. And that is the cup that's going to present the antigen to the immune system. All right, that's all we have time for today. Please read this over because we'll jump straight into the mechanism of antigen presentation. Guys, I know this is complicated. I know this is difficult. Spend some time with it. That's the only way you'll get comfortable with it, is by spending time with it. There's no easy way to present the immune system, because the immune system is not simple. So formulate some questions, ask some questions. Don't be afraid to ask questions. Your lab books, Mrs. Schaefer has your lab books. So go see Mrs. Schaefer for your lab books. Some of you still need to turn lab books in. The rest of you, I'll see either Tuesday or Wednesday. Those of you on Zoom, please get better soon. All right?